I knew we should have done a karaoke session beforehand just to check out the microphones. I want to thank you all for joining us this evening. I don't know about you, but it's actually my first With Insight where there's been daylight outside, and it's so refreshing. It feels very spring-like. Welcome to With Insight, and welcome to everybody that's here in person. Thank you for joining us. And I also want to thank the folks who are joining us online. I do want to remind folks online that you are welcome to interact with us. Please use the Q&A function. Please feel free to ask questions as we go through the session so we can pick them all up at the end. So I'm going to start off with a very brief introduction of myself. I'm Jane Wilkinson. I'm the Executive Director of the Koch Institute. I joined the Koch last summer. We were still very much in lockdown then, and I remember letting myself into the building and being the only person here. So being here and kind of talking about our COVID research and how we've been moving that forward is kind of very close to my heart, having joined here right in the middle of it. This session tonight is about revisiting the work that my colleagues Hojan, Angie, and Mike did during the pandemic, and how the Koch decided to really take part in pivoting to, our cancer research towards COVID research. As you all know, the pandemic started 2 years ago, and I just want to take a quick moment to recognize and thank our physicians, our clinicians, and our caregivers who were on the front line and are also still very much on the front line. So on behalf of you all, I would like to thank you, especially our clinicians and who are presenting to us tonight. And I also want to take a moment to recognize the tremendous amount of pain and loss that we've all been through over the past 2 years. But what I want to do is think of this event as a way for us to reunite and come back together again and acknowledge the resilience and the importance of science and scientific research, and how important it's been through the pandemic. So tonight we're going to be talking about 3 different topics, which I think very much kind of exemplify everything that we were thinking about with COVID and with the pandemic. We're going to be talking about personal protective equipment, how are the frontline carers taking care of themselves. We're going to talk about testing, looking at immunity and looking at antibodies. And then the last talk tonight is actually going to be thinking about therapeutics. So really kind of the 3 important parts of how we got through the pandemic and how we're still very much going through it, especially as we hopefully move to more of an endemic phase. So my colleagues tonight, Angela Belcher, she's a James Mason Crafts professor, and she's also the head of the Department of Biological Engineering. We have Hojan Lee, the Charles and Jennifer Johnson clinical investigator. And lastly, we have Mike Yaffe, who's a David H. Koch professor of science. He's an intensive care physician, and he's also the director of surgical oncology for NCI. So instead of doing incremental introductions for everybody, I'm going to let our presenters go through their talks. I'll be able to take 1 or 2 questions after each talk. And then at the end, I'd like to bring everybody together and have more of a panel discussion and really kind of talk through how we got to where we are now and maybe kind of look towards the future as well. So with that, I'm going to hand over to Angie.
Thank you so much. It's incredible to be here in person, and I'd also like to acknowledge the people online. Thank you for being here. It's just amazing to be back, full time in the Koch. This is such a home for me. It's an amazing interdisciplinary place where it's just so great to be a part of, and being back with the students and the faculty and staff and postdocs. It's a great feeling. So I'm going to tell you a little bit about how things shifted for us during the pandemic. So I'm really a solid state chemist by training. I'm a materials engineer. I build things, but I build things with biology, and I like to build things with viruses. Actually, M13 bacteriophage, which is a beautiful bacteriophage that only infects bacteria. I'll show you a cartoon picture of it in a minute. But I've spent my career, it's really my amazing lab and students. I've spent my career engineering M13 bacteriophage to make things that are useful. And so the kinds of things that we've made are batteries. So these are virus-based batteries, virus-based solar cells, virus-based catalysts, viruses that can find tumors. This would be a tumor that a size you could maybe find with a CT. And these are down here a tumor that you can find with our materials we engineered with viruses. And it's just been really fantastic to be able to work in that area. Right before the pandemic, actually, we started thinking about making high surface area carbons for actually battery electrodes, and then pretty quickly shifted to use those high surface area carbons for absorbent materials for many different applications, including toxins and very toxic industrial chemicals, and then started shifting to make PPEs for trying to protect against SARS-CoV-2. And so basically what my lab does is we study how nature normally makes materials, and we like to, which in the case of shells and glassy sea structures, is to take ions in solution and use proteins to grab onto those ions and start building materials. Now, organisms have been doing this for a very long time during this red arrow, the Cambrian geologic time period. And what we like to do is repurpose bacteria and viruses to mimic some of these processes, but instead of building shells and things, we like to build solar cells and batteries and materials for cancer. And so we like to give biology the ability to work the whole periodic table to be able to evolve from that. And so we work in energy, healthcare, and the environment, and we've spun out many companies in these areas. And to us, it's really controlling the atoms and being able to control them to have many different kinds of applications. So here's our M13 bacteriophage is this beautiful virus. It's long and skinny. We genetically program it to grow all these kinds of materials that you see here. These are normally not materials biology would grow, but we encourage it through DNA changes and through protein changes to grow that. There we are, happily growing our materials. We started growing these materials that are carbon-based materials, really high surface area carbons. And this is one of the things that we did during the pandemic is we took viruses and we programmed them to grow really high surface area carbons to be able to neutralize toxic materials. And so we're interested in all kinds of toxic materials from toxic materials in coming out of industrial processes to rivers and lakes, to pollution, to if we dropped it, it would go everywhere. It's so high surface area. So I'll pass these around. This is our carbon-based materials that were woven into textiles to use for masks. So what we basically did was normally people make carbon fibers using big expensive machines and high voltage materials, but we wanted to make high surface area carbons just in a beaker, genetically templating with viruses. And we basically took some chemistry that's really old chemistry. This is called phenolic resins, polycondensation. You've used these kinds of resins in many parts of your life. But what we decided is if we could get viruses to make them for us, we could make them nanostructured. We could really control their architecture. By really controlling their architecture, we could give them a lot of surface area and a lot of reactivity. And actually, the surface area of the materials that's going around, the reason I said not to drop this is because it's very light and it would fill up everything. But there it goes. Okay. So, but the surface area of these virus-based high surface area carbons we made are on the order of 2,000 meters squared per gram. So that's taking 1.5 hockey rinks, the surface of that, and putting it into 1 gram of material, and that's the surface area. Now we control the porosity, we control all the chemistry on the surface, and we can put different kinds of materials, aluminum, iron, many different kinds of materials on it, and do wonderful things with it. So we first started doing this is pre-pandemic. We actually found that it was really good at absorbing toxic dye molecules, heavy metals, We started looking at drugs for possible drug overdoses. And what we noticed about it is the kinetics, how rapidly it took it up was about 30 times faster than benchmarking against commercial materials. And so what I said was, wow, what molecule would you want to take up faster than any other molecule if you could? And no one else really came up with this answer, but I said I'd actually want it to be chemical warfare agents. I'd want it to be sarin, and mustard gas. And so we started actually working with the Department of Defense and sending our really high surface area materials to them along with other toxic industrial chemicals. And they said, yep, yours works better. Yours takes it up very quickly, but another thing that yours does that others don't is yours breaks it down into non-toxic materials. So you can take something very, very toxic like sarin or soman, and it gets broken down into non-toxic chemicals. And this was all done through genetic engineering viruses and also other kinds of materials. And not only that, we could actually break it down almost 100% with just these genetically engineered materials. And you could actually put them into a mask and you could still break them down instead of having to use a really gas mask that you might use if you're and so we also found that the breakthrough, this is actually looking at the breakthrough, this is the blue is a control material, a commercial material, and here's ours. So this is the amount of time it takes, you can be exposed to it until it starts breaking through your mask or other kind of material. So ours are definitely superior. And then we started saying, wow, what if we could do this for the next flu? And then our lab shuts down. And then we were going, yeah, we had this idea of maybe we could use this mask material that we're using. We thought, we put what we thought masks looked like at the time. Of course, we're all very familiar with masks now. And we started saying, okay, well, we could actually make these into textiles. We can make them into wipes. You can make them into sensors. And when you make them into these textiles, they still have really good properties against all the chemicals that we were testing them against. And we said, and we actually worked on scaling these up to a really large scale during the pandemic with the Army lab. So we went from 10-liter reactors to 1,000-liter reactors during the pandemic to scale up these materials. And so we started testing them through small viruses, MS2. These are not infectious to humans, but they're about the same size as SARS-CoV-2. And what we found is we could find formulations that would completely filter out the same size viruses. So this is looking at what the normal concentration of the virus is, and you try to pass it through a filter. We're able to completely filter it out with a couple of our different formulations, which we're really, really excited about. But we said, okay, well, what if could you actually deactivate them as well? And sure enough, we found that 2 formulations out of the 30 or so that we made could completely deactivate MS2 virus. And so it wouldn't be able to infect, in this case, its bacterial host. And so we said, okay, what about SARS-CoV-2 or a close surrogate of that? We can't work with those in my lab, so we sent them out to the Army and tested the ability of our materials to inactivate that. And we got near complete deactivation as well with our materials. And so that's what we developed. We would have never done that if we didn't need to. We also then put magnetic materials in them. Now we can magnetically separate out viruses. We showed that you could do bacteria as well. We said, why do Clorox wipes work really well? What if we went in and genetically engineered our viruses to also have similar kinds of things? Could we go and inactivate bacteria? Yes, we could. We could deactivate E. coli. Again, filter out and deactivate E. coli using these materials that we made. So where are we going from here? Well, we now made one platform that does toxic industrial chemicals, viruses, chemical warfare agents, E. coli, and now in the future we're working on PFAS and other forever chemicals that are fluorinated chemicals in water, and that is where we're heading now. And our initial data says that actually we're doing quite well at filtering out some of the most toxic fluorinated carbons that are found in waters, and ours are outperforming the standards. Where are we going from there? What we really hope to do is now break these materials down the same way that we were able to prevent viruses from infecting, breaking down chemical warfare agents. So that is basically to tell you how the mind of a solid state chemist goes from batteries to cancer to working in the pandemic to coming out of the pandemic, hopefully soon, and starting to think about other kinds of environmental contaminations that are a problem in our environment. So thank you.
We have time for one quick question, if anybody has one from the audience or on Zoom. If not, we'll keep going. Oh, we do have one. Great.
So in the carbon emission there may be some legal from the
Angie, we got one question coming in over Zoom. Does the protective effect degrade with time and the quantity of exposure?
That's a great question. So if you're talking about whether the effect of the degrading happens over time, changes over time, it doesn't change in the powdered form. I know that for sure. Like the powder that we have, we're actually developing these for shampoos and other kind of wipe down textiles as well. But we don't know as much in the mask. It's a great question. Our work is a little less advanced in getting the formulation the best for the mask, but obviously that's going to be very important. But it is a it's a solid material. It's not a polymer. It's not a biological material at this point. It is. I imagine that it will stay active for years.
Right. So first off, I'd like to thank everybody who's here in attendance as well as online on Zoom. It's really exciting to be taking part in this as we, as Jane said, hopefully moving into past the pandemic phase. I'd like to take this opportunity to tell everybody or give everybody an update on our work on trying to figure out a way in which we can actually have bedside or at-home testing to quantify how much protective immunity individuals have against SARS-CoV-2. I myself am not trained necessarily as an engineer. I'm actually an oncologist and bone marrow transplanter at Boston Children's Hospital and Dana-Farber. And so it's a bit of a roundabout way in which my lab that studies leukemia and blood cells stumbled onto this, but it's something that is very important to us now. When you feel the call of duty with a pandemic, you go where the science takes you. So before I start, I will disclose that MIT has filed several patents on this technology, for which I'm an inventor. But the even more important thing to disclose is that none of this project would have been possible without the collaborative spirit here at the Koch Institute and at MIT. We have never, in my lab, done any protein biochemistry work, never done any diagnostics or specifically paper-based diagnostics. And if it wasn't for all the tremendous collaborators that we had here, none of this would have even gotten off the ground. So I do want to acknowledge that. And especially, I do also want to acknowledge the two members of my lab who really were the ones that drove this work forward from our lab's perspective, which are Guinevere Connolly and Orville Kirkland. So in thinking about how our own bodies respond to viruses, it's really the immune response that governs to what degree we are protected from any type of pathogen. But in this case, we're going to focus on SARS-CoV-2. And so our immune system certainly has an innate aspect where there's this baseline low level of defense that can respond non-specifically to whatever is sensed as an invader or non-self. But what's really fascinating about our immune systems is that it can actually adapt and recognize when pathogens have tried to attack the body before and store that information, have a stronger response if they ever try and come back to attack our bodies, which is incredibly relevant right now for how we know that SARS-CoV-2 reinfections can happen, or infections even after immunizations have happened. And so the fascinating parts of this adaptive immune system are really driven by two general arms. And so this is sort of broad strokes generalization. But in adaptive immunity, there's the cellular arm, which is composed of T cells killing off cells that have been infected by viruses so that those infected cells can no longer be factories for making more virus to spread through the body. And there's also the humoral arm of the adaptive immune system, which is predominantly driven by B cells, which produce antibodies. And these antibodies can actually bind to viruses and neutralize them from being able to infect subsequent cells. And so when trying to figure out how much of each of these arms is actually active at a given time, it's incredibly hard to measure how much cellular immunity that you actually have. And I think the immunologists in the room in this building can tell you that we have to develop extremely advanced assays to detect this. But antibodies from the humoral arm are actually much easier to measure. Like, we've been measuring antibody levels for decades upon decades in medicine. So this is very easy to do. And one particular class of antibodies that we are very interested in are neutralizing antibodies, because these are the antibodies that will actually prevent a virus from infecting any cells that it is around. So they prevent SARS-CoV-2 from actually entering human cells. But the problem with neutralizing antibodies, whether you've been exposed to natural COVID infection or if you've been vaccinated against COVID, is that they decrease over time. And so numerous medical publications have demonstrated this, but as you get further out in terms of months from when you received the vaccination, whether it was the Pfizer-BioNTech or the Moderna version of the mRNA vaccines, you are losing your neutralizing antibodies, no matter what type of assay you run for neutralizing antibodies. And what goes hand in hand with this is the risk of reinfection also increases the further out you get, such that multiple different studies have now shown that although there are many aspects of the adaptive immune system, the number one correlate of protection against SARS-CoV-2 infection that we can actually measure is neutralizing antibody titer. But the problem with measuring neutralizing antibody titer, because I'm sure everybody in this room would want to know what their neutralizing titer is, all of the assays to date are extremely expensive and take a long time to run, ranging from several hours up to several days to actually run, plus all of the reagents that are required for them. And so we thought this was a problem that maybe we can potentially work on, and we have an idea for it. But what really drove us to really sort of grab hold of the reins and aggressively pursue this is that there are several vulnerable populations where this is incredibly important for. And these are populations where the cellular immune system we know is suppressed. And those would be near and dear to my heart: cancer patients who are receiving chemotherapy or receive bone marrow transplants, but also a variety of different people who are immunocompromised from having autoimmune disorders, or even just the elderly, where we know that T cell function is suppressed. So if we don't know how good your T cell function and cellular immunity is against protecting from COVID, then we darn well better want to measure how well your antibody levels are protecting you against COVID. And if we could do that, then we could give these vulnerable populations a sense of how well they are actually, what risks they have. So when trying to attempt to develop a way to measure neutralizing antibodies, and our goal was to maybe even do this in a way that you could do this at home or just walk into a CVS and get an on-site test, looking at how neutralizing antibodies work, what they actually do is they can bind to the surface of the virus here and then limit its ability for the receptor binding domain of the spike protein on the surface of SARS-CoV-2, prevent that from actually binding to the ACE2 receptor that's on the surface of cells that's trying to infect, such that if you're covered with these neutralizing antibodies, you can't infect a cell. And so we thought this relationship, neutralizing antibodies binding to spike and preventing spike from binding to ACE2, that's what we would need to test. And so we came up with this particular schematic or strategy here, where we could take a drop of patient blood, drop it into a tiny little tube that has some SARS-CoV-2 receptor binding domain in it, mix it up, apply it onto a strip that look very much, for all intents and purposes, looks like a home pregnancy test, except it's a drop of blood and some buffer that goes on there instead of urine. And then the solution would then flow down this line or down this piece of paper that's on the inside. And what would then happen is we have one line here where there is ACE2 protein that's been spotted onto and dried onto the piece of paper here. And if any receptor binding domain is not neutralized, i.e. doesn't have neutralizing antibody bound to it, it will actually be able to bind here and then sort of just get stuck. And this receptor binding domain has a little bit of colloidal gold in it, which will give it a red color. And so you'll see a red line appear with RBD that's not neutralized and can bind to ACE2. But if the RBD molecule is neutralized by this antibody that looks like a little Y right here, that RBD is no longer able to bind to ACE2 and will just flow on past. And then we can capture these molecules that flow on past by using protein A right here, which is a bacterial protein that pretty much can bind to almost any antibody out there. And so these neutralizing antibodies that have neutralized the RBD, they will then be captured here. And by extension, that colloidal gold red signal will then show up here. Okay, so that was the basics of how our assay worked. And so in practice, when we constructed these, we were able to take advantage of some clinical collaborations to get serial samples from some patients who had blood collected the morning before vaccination and then at various time points through post-vaccination courses. And so this is somebody who received the Pfizer-BioNTech mRNA vaccine. And so on the day of vaccination, you can see that this infectious line here, and we call this infectious line because the RBD is still infectious because it can bind to ACE2. So at that infectious line, we see a signal here, but there's clearly not going to be any neutralizing antibodies for this patient who never had COVID and is just getting immunized. And so at the neutralized line, where RBD has been neutralized by neutralizing antibodies, we don't see any signal. But as we progress through and sort of peaking about a week or two after the second dose of the Pfizer vaccine, we see a nice strong line appear here. But we also see that signal intensity decrease over time. And so this is a nice qualitative signal, but with neutralizing antibody titer, you actually want to measure a number because the relative titer or amount quantity of those neutralizing antibodies dictates what your protection is. And so we wanted to be able to generate a number. And so we could then quantify these pixel density at each of these individual lines. And what was really important to us here is that for anybody who's ever run any lab assay ever, from day to day when you run it, there are variations. And so that signal line intensity could vary, but we had a nice internal control here in terms of the infectious line that would always appear at relatively the same intensity. And so if you can then normalize the signal here at the neutralized line divided by the signal at the infectious line, then you could have an internal control that you can compare day to day, week to week, person to person, et cetera, et cetera. And so what happens then is we also took a bunch of samples from post-Pfizer, post-Moderna vaccinations, quantified this neutralized over infectious line ratio, and compared it to the gold standard viral neutralization assays, and show that the kinetics of how your neutralizing antibody titer in our assay goes over the course post-vaccination is very similar to what the gold standard assays show. And then finally, we wanted to start to try and see whether or not we could have good sensitivity and specificity for detecting specific titers of neutralizing antibodies. And so here, what we're doing is the gold standard in clinical pathology labs of called receiver operating characteristic area under the curve analyses. And basically, we're just setting a bunch of different thresholds for N over I ratio and seeing at those different thresholds, what is our sensitivity and specificity for identifying, and this example here is a neutralizing antibody titer of 1 to 100, which is a pretty decent titer, but not super high. And what you want to see in a high-performing test is that you want this red line, the corner of the red line, to get as close to this upper left-hand corner as possible. The closer you get, the higher performing your test is, i.e. better sensitivity, better specificity. And so we see a pretty excellent area under the curve for the aficionados here. And you can use this line to then determine, okay, at the very peak point here, we have a 91% sensitivity and a 91% specificity for saying whether or not an individual has a neutralizing antibody titer over 1 to 100 or less than 1 to 100. And you can do this then for a variety of different titers, because we actually in the medical field don't know yet what the true protective neutralizing antibody titer is. And so over a whole range of different titers, we see that we have fairly excellent area under the curves. And what we also compared is that the previous best standard for measuring antibody levels to SARS-CoV-2 was to do a ELISA test to detect all IgG antibodies against spike proteins. And we see that at the lower antibody titers, we are approximately similar to that. But really, when we're talking about high antibody titers, excellent protection, this at-home test far outpaces a lab-based ELISA for total spike protein IgG antibody. So we were excited about these results and are trying to move forward with industry collaborators on actually getting this onto the market. But we think that the particular use cases where this is going to be very valuable is to serially monitor for when you actually need a vaccine booster, when you need to get back up to those higher 1 to 500, 1 to 1,000 neutralizing antibody titers. And this is especially important for vulnerable populations where we know that we can't necessarily trust the T-cell cellular immunity. And in addition, I had also mentioned that there was the problem where we don't know what the true exact level of neutralizing antibody titer that is protective against infection is. And part of that is the fact that all the other neutralizing antibody assays that can be run are very, very expensive, resource intensive, take very long. But with this type of an assay, you can run this on thousands, tens of thousands of people in a serial study to see at what point people actually lose protection at what certain degree of neutralizing antibody titer. So I'll wrap up there and acknowledge that we have some other collaborators that were outside of MIT that we'll acknowledge. Tyler was really a big champion of this project when he first heard me talk about it back in March of 2020, and really encouraged me to go forward this. And then I will also acknowledge Lisa Marks Schwartz, who was really instrumental in being able to get us funding to actually pursue this. So I'll wrap up there.
Time for a quick question. Angie.
Just one time. Yeah, that's the goal.
And Hoshan, just a question for me. What, when we talk about how expensive these antibody tests are, I remember I took one personally and I paid for it out of pocket, and it's $100 and something. And it took weeks to come back. What is the price point here that you're hoping for?
Yeah, so I guess I don't really have a whole heck of a lot of experience in industry and biopharma, et cetera. But in terms of raw materials, we're talking about, I don't know, maybe less than $1, certainly less than $2. And it runs, I mean, we've done various time courses for how long it takes to run. It could be 10 minutes, 20 minutes, something like that. Yeah.
Thank you.
All right, I guess I'm the wrap up. So thank you all for your attention. I'm Mike Yaffe, and I'm going to tell you a little bit about what happens if the PPE that you get from Angie doesn't work so well, and you're unfortunate enough that the antibody titer test from Hojan says you don't have good antibodies and you have the great misfortune to come down with COVID and develop respiratory failure. So I want to tell you an update on an old drug for a new disease. So we all know that COVID was this horrific pandemic. And for the first 2 years or so that it was out, we really had almost nothing we could do because there was no vaccine, there was no effective treatment, really wasn't clear what to do. And, I wear a couple different hats. One of the hats I wear obviously is as a scientist focused on cancer and cancer biology. But I also am a surgeon and an intensivist. And we began to see at one of the hospitals here in Boston, we began to see our ICU fill up with patients with respiratory failure. But there was something very funny about this respiratory failure. First, we began to get in a situation where we were running out of ventilators. We were using the ventilator that we use in the operating room, and we weren't operating anymore. We were just putting patients in the operating room because we didn't have any more ICU beds. And it became very scary as we began to contemplate what were we going to do when we ran out of ventilators. And the pneumonia that we saw looked very much like the type of pneumonia that we were used to seeing previously in a lot of patients that have something called adult respiratory distress syndrome. But there was something that was very different about this pneumonia. First, a patient could come in and sometimes sometimes their lungs would look like this, and they actually weren't symptomatic. They weren't complaining of being short of breath. And then within 12 to 24 hours, they would be on a ventilator on 100% oxygen. And 24 hours later, they'd be dead. We'd never seen anything like that. And there was something else that was very different about this type of pneumonia that made it different than the usual type of pneumonia that we see in adult respiratory distress syndrome. One of the things that was funny about it was the way the lungs behaved on the ventilator. So ordinarily, when we put someone on a ventilator, we dial up their oxygen, and oftentimes, as their lungs fill up with bacteria and fluid, we have to use higher and higher pressures to be able to inflate their lungs. And you can think of the lungs, in essence, as a spring and a dashpot, a spring and a shock absorber. And as the lungs fill up with fluid, the shock absorber gets stiffer and stiffer and stiffer, and we need higher and higher and higher pressures. But this wasn't the case. COVID-19 respiratory failure, boy, the lungs were very compliant. The lungs behaved, almost like normal lungs, and yet these patients were not moving oxygen into their bloodstream very effectively. What in the world was going on? And the clue actually came from the discovery that these same patients that we were seeing with pneumonia, a subset of them actually were dying of heart attacks, and that a different subset of them were presenting with strokes. Young people with strokes and heart attacks. We'd never seen anything like this. And what it began to suggest was that the problem, in fact, was not a fundamental problem with the fluid filling up in the lungs. It might be a problem with blood clots. Some sort of blood clotting complication was killing our coronavirus patients. Now, I have to tell you, the part of my lab that studies tumor biology is very interested in trying to understand the interaction between the tumor microenvironment and the tumor. And in particular, part of the lab has been working on how inflammation and blood clotting control how tumors behave. And that's because we know that one of the things that malignant tumors have a tendency to do is cause blood clots. In fact, some patients present with cancer initially because they have an unexpected blood clot in a leg or their lungs. And in the workup for this, we discovered they have cancer. And in fact, it was turning out that this process that we had studied in the lab for cancer seemed to play a role in COVID. It looked like we were getting a process called immunothrombosis that had really not been well appreciated before, where the viral infection was causing inflammation. And one hallmark of that inflammation was blood clotting within the blood vessels through which oxygen in the lungs is transmitted into red blood cells. So I'm going to show you some slides. I apologize if they're a little gory. These are autopsy specimens of lungs from patients in China. And what I'm pointing out to you in the lungs is stuff like this and this and this, where the blood vessels in the lungs are filled with blood clots. We saw it in China. Whoop, did I skip Brazil? No, I guess not. We saw it in the specimens from Italy, where we again saw lots of evidence of blood clotting within these lungs. And we saw it as well in Brazil, where you could begin to see these micro clots within the very small blood vessels lining the lung. And this suggested to us that maybe part of the problem was an imbalance between clot formation and clot lysis. Now, we're all familiar with blood clotting. We cut ourselves on an envelope, or we skin our knee. And that process, of course, initiates blood clotting, where we use thrombin to convert fibrinogen into a fibrin blood clot. But we have to limit that. Otherwise, every time you cut yourself, you would clot all the blood in your body and you would become one complete clot. And so that's normally limited because you have a system in your body that performs a process called fibrinolysis. That's where, at the same time that you're forming a blood clot, you're lysing that blood clot to limit its size and limit its growth. And that involves another enzyme called plasmin that breaks down that fibrin blood clot. And plasmin in our body is normally not present. It's formed from a precursor called plasminogen through something called TPA. You've probably heard of TPA. This is what we give people with strokes and heart attacks because it activates plasminogen to plasmin and lyses the clot that's causing their heart attack or their stroke. And ordinarily, this is controlled by another inhibitor called PI1. And we reasoned that maybe there was some dysregulation between blood clotting and between blood clot formation and blood clot lysis that was underlying what we were seeing. In fact, we knew some of this from work in our own lab. We knew that in trauma patients, for example, who bled to death, they actually had too much of this TPA. they were making too much plasmin and lysing too many blood clots. We knew that another subset of those patients actually were forming blood clots just like the COVID patients. And although they had a lot of TPA, they also had even more of that inhibitor. And we reasoned that maybe there was some imbalance in COVID patients between the amount of TPA they were generating and the amount of this inhibitor that they were forming. And so we reasoned that maybe we could just treat these patients by giving them TPA, just like we would give TPA to patients who were having heart attacks and strokes. And so we wrote a proposition paper early on and said, is there a role for tissue plasminogen activator, TPA, which we're already using, as a novel treatment for refractory COVID-19? And boy, did we get some pushback for that, because people didn't like this idea of lysing blood clots in people that were critically ill. And we know that if you do this in patients who have strokes or heart attacks, there's some incidents with which they just bleed too much. They can bleed into their brain. They can get an intracranial hemorrhage. That's the big risk factor here. And so we proposed to test this, and using money that was generously donated to MIT and a deal we made with Genentech, we performed what was called the STARS trial. This stands for study of alteplase, that's TPA, and respiratory failure and survival. And we hypothesized that maybe we could give TPA to patients with COVID-19 who had severe ARDS and were dying in front of us and not responding to anything we could do. And maybe we could get them well, maybe we could get them off the ventilators, and then we'd have more ventilators to use if we became overrun. And so we started a trial to take patients 18 to 75 years of age and give them TPA if they were failing with respiratory failure so severe that there was nothing else that we could offer them. We started with 6 patients. We eventually, I'll show you, increased this trial to 50. We sipped going on and done another 80, and I'll show you what the results were. So this was published about a year ago. I have to really thank Chris Barrett. This was a resident and a postdoctoral fellow in my lab here at MIT who really drove most of this study. And what we did in this study was we took 365 patients, of which we could find 50 who we could randomize, 50 who were dying in front of our eyes. They had no other options for therapy. And we split them. 25 of them were controls. We treated them just like we would treat everybody else. And 25, we either gave 1 shot of TPA or we gave them a shot of TPA and later on put them on a TPA drip. Now, interestingly, because therapy was changing so rapidly as we were doing this, we had to create a rapid, adaptive, pragmatic, randomized trial. I have never done a clinical trial like this before. This is a trial that you learn as you do it, right? To try to make it better, and you have to adapt to the new therapies that are coming online. I just have to tell you one more piece of data, and that's how we're going to evaluate respiratory function. In you and I, we breathe room air. That's 21% oxygen. And the amount of oxygen that's dissolved in our blood is about 90 millimeters of mercury. And so we can define what we're going to call the P to F ratio. That's the PO2, 97 or 90, divided by the FiO2, that's 0.21, to get a number of about 460. And all of us sitting here today probably have a P to F ratio that's over 400, which is normal. Below 400, you're not getting enough oxygen in your blood. And below 300, we call that respiratory failure. Anything we can do to get that number higher, even if it's really severe respiratory failure, the higher we can raise that number, the better the patients do. Okay, so here's what the first piece of data showed us. The first piece of data showed us, if we looked at that P to F ratio between the control group and the people that got the TPA, we already saw some benefit that was statistically significant after just 2 days. In fact, if we made our criteria, not that their P to F had to be super, but it had to at least double. In fact, only 2 patients in the control group were able to do that, 11%, whereas almost half of the patients we gave TPA to were able to improve their oxygenation significantly. This is what their P to F ratio looks like. The control patients didn't get better. The patients that we gave our TPA did, you can see here, definitely showed an improvement. More importantly, during that period of time, if we looked at their mortality, the control group is in red and the people who we gave that TPA bolus to are in blue. Boy, we saved some lives of people that would have otherwise died. If we added to that a TPA drip, they actually got worse. So we quickly abandoned that. It was an adaptive trial. We stopped doing that. Okay. And then here I just want to point out, this is ventilator-free days. So the controls had no days they were off the ventilators. Our patients had 12 days out of their hospital stay they were off the ventilator. There was no difference in the amount of time they were in the intensive care unit, but they had 6 more days of being out of the intensive care unit during the course of their hospitalization. Finally, if we looked at the incidence of bleeding, it was roughly equal between the groups. We didn't have bleeding complications here. Now, since then, we've gone on and gotten another 78 patients not in a trial who we could retrospectively evaluate. This was called the MUST trial for multicenter study of tissue plasminogen activator in COVID. Here, we were able to get 80 patients, 33 of whom survived, 46 of whom did not. And so this is a retrospective trial. It's not as powerful. But the thing we noticed was that if we split the patients by how they were doing, those patients who were doing okay, this is again a measure of that respiratory failure. Patients who were getting better at the time they got TPA continued to get better, but we didn't make much difference. The people that mattered were the people that were going right down the toilet, who were destined to die. And if we could give those patients TPA, they were the people who got better. Okay? And again, when we looked at the complications from doing this, I just want to point out we had very little bleeding episodes, and we had 1 incidence of an intracranial hemorrhage, 1%. That's lower than what we see if we give TPA for strokes or for heart attacks. So what I've told you about today is that TPA treatment is an effective therapy. If you have severe COVID-induced respiratory failure, and we can't save you by using any other treatment, that the treatment actually seems to work best in patients who are destined to die, those people whose clinical trajectory is actively and progressively going downhill. And it seems to have a very acceptable safety profile with really minimal risk of clinically significant bleeding or bleeding inside or bleeding into the brain. And I think what we've learned from this is, while now this is potentially not so necessary, given what we now know and have in the way of vaccines, when the next wave of COVID hits and we're in trouble, or another non-COVID pathogen emerges that has the same immunothrombotic biology, I think we now have a new tool in our armamentarium. I'll stop there and be happy to take any questions.
Mike, one quick question before we have a quick panel discussion, and this has just come in over Zoom. Your TPA group indicates that you added heparin. Do you know that the TPA was necessary and that the effect wasn't just due to adding the heparin alone?
Yes, an excellent question, and I should have been clear. All of the patients in the control group and in the TPA group received heparin. In fact, one of the things that came out of this work, together with which was really led by a group in Pittsburgh, was the discovery that heparin itself is of benefit, but not if you're in the ICU. By the time you're sick enough to be in the ICU, it's too late for heparin. But those patients who are not on heparin and are going downhill with COVID, but not yet in the ICU, they're the people we ought to heparinize. Excellent question. Thank you.
Thank you. I'm just going to ask the panelists to join us for a couple of minutes of a discussion. Hey, we see faces. Hi,
Yeah.
everybody. So my first question to you all, here we are 2 years later. We've gone through a complete lockdown. We had our labs closed for a period of time. We reopened, we pivoted, to think about how we can move our research here into more COVID-specific projects. How are you managing the pivot back? How have you managed to pivot back? And how's that adaptability of us thinking about COVID and then COVID and cancer back again? How has that affected how your labs operate and how you all think about problems moving forward?
Yeah, so I guess I can go first, but we never actually really used to think a lot about diagnostics at all. It was kind of interesting. We were much more mechanisms of leukemia, blood cell formation, but seeing the power of how a sort of a real-time diagnostic could actually be of great utility. We've since moved back to our normal mechanistic research, but we've actually picked up an entire arm of our lab that's focused on blood-based diagnostics. And one of the great things about being in this environment of the Koch and being at MIT, there are just so many smart people with good ideas. People recognize we are a lab that specializes in blood cell-based research, but now we have this whole diagnostics component, and we have all these students coming saying, hey, these are two things I'm interested in. I have this idea or that idea, and it's just this almost an avalanche of really interesting new projects that are being proposed, and that we're pursuing some of them with regards to that.
Well, it's amazing to be back, and it's amazing to be back working on cancer, really. I couldn't be any more excited about that. So that pivot back has been very important to our group. We're engineers in my group, so we solve problems. And so when Susan Hockfield first asked us to work on cancer, we're like, no, we're not going to work on cancer. We work on batteries and solar cells and catalysts. And opening that mindset of thinking about, okay, what are we good at? What can we take what we're good at and apply it to problems we haven't thought about before? That gave us a tremendous amount of confidence, actually, to be able to pivot backwards in different directions. And so pivoting towards working on COVID was a good extension for us. It's allowed us to think about other viruses, and then really start to really think deeply about environmental impacts as well, which is a major area that we're interested in right now. But ultimately, at the end of the day, we try to look at what our skill sets are, what are we good at, and how can we apply those to important problems in the world. So we'll continue to do that.
I guess first, I would certainly agree. It's great to be back. As a surgeon, I think for many years, we were a voice in the wilderness, crying about blood clotting and its interaction with the immune system and how that might play a role in cancer. And suddenly it sort of became mainstream after it was realized that there was this link between inflammation and blood clotting in COVID. So that clearly has, I think, helped to spread the word that that all of the systems are interconnected. The clotting system, the innate immune system, the adaptive immune system, and our natural defenses against infection actually are co-opted by cancer. And so this has sort of cross-pollinated the two parts of my lab, part of which has been focused on inflammation and blood clotting, and one part of which has been really focused on cancer therapeutics, to realize that we really need to be talking to each other a lot more. And that's been a real shot in the arm, I think.
So COVID is still very much with us. And I do want to recognize it's been a very, very tough time for a lot of us, and it still is a journey that we're on. But I think for me personally, it has shown me the resilience of science, and and if anything, I think we'll all agree that science kind of got really, really cool during COVID as well. But also, the importance of collaboration, and especially if we think more broadly around the vaccine development programs, which were global collaborative efforts. What has COVID showed us about the importance of our collaboration here? And for those in the audience who aren't familiar with how we're organized, physically, we share each floor here in the building with engineering and biology and chemistry. Mike mentioned cross-pollination. We mingle and mix everybody up on each floor to make that collaboration happen. Collaboration, I'm sure, took a hit when we were remote, we were on Zoom, we were hybrid. How are we bringing it all back together again?
I think we're seeing a resurgence as we begin to come back of this interaction. I think the one thing that COVID did was it made us realize how we had to work together. And in a way, I think the fact that we became interdependent on adjacent laboratories because we could only have a certain number of people in the building at any one point in time sort of forced some communication between labs that might otherwise not have happened. So I think that's been really a strong and positive feature. And I think the need to be able to translate what we see in the lab back into the clinic as quickly as possible has also, in many ways, really reinforced the need to collaborate, because in our lab, which mainly does very basic science, right, we had not been nearly as actively engaged with the engineers as we are today. I'd say this has definitely helped to push things in that direction.
You are an engineer.
I am an engineer, it's true.
You're my
I
department,
was a
know
material
that.
scientist in her field, but I'm embarrassed to say it because I'm so dwarfed by the things that Angie has done.
So I also agree that that labs did get physically separated during the pandemic, and it is true that we did have to share spaces based on the dedensifying and then the missing reagents during the supply chain issues. And
I know there was a trade-off on who had the most tips at one point.
right, yeah. But I mean, thinking about it now, so collaboration is such a huge part of our work, and I had names for my Army collaborators listed on the slides, but Greg Peterson and Jared DeCoste, who were amazing collaborators at the Army. We grew more collaborators a long distance during the pandemic, and that includes other hospitals and research labs around the country, because part of it was Zoom initiated, because Zoom just got to be such a thing that it was just normal to have these Zoom meetings, that I'd say our collaboration has just really expanded during the pandemic. But that part's been amazing. But the sharing the bench and sharing the conference room did suffer, but it's starting to come back here. And I really think, although I said, I'm an engineer, in this building, it doesn't matter if you're a scientist or you're an engineer, or because we all have a common goal, and we all bring the best of what we do together to solve that common goal. And that probably was missing some, but I think it's starting to come back again.
Yeah, I mean, even for us, particularly with this project in the depths, the initial of COVID, that collaborative spirit here, which I had mentioned, it was just amazing, because I said we knew how to do none of these techniques, and so we went to people in Mike's lab, Angela Kohler's lab, Sangeeta Bhatta's lab, Hadley Sykes's lab, and we basically said, hey, can you teach us how to do these things? We have this idea. And they said, not only will we teach you, we will work on this with you. This is super important. And seeing how every scientist just seems to rise to the occasion if there's something really important, that's really a beautiful thing to see and have reaffirmed. But I'll also second that the rise of Zoom or any virtual collaborations has been tremendous. I honestly did not know how to use Zoom when I first became a PI. The first lab member that I ever had to interview solely by Zoom is sitting there in the back, and it was a disaster with how I was operating Zoom, but I'm glad that we still were able to hire her. But you can literally reach out to anybody in the world. And previously you would say, okay, yeah, 6 months from now, we'll meet up at this meeting and talk about what we're going to do. But now it's, tomorrow? Oh, I got an hour free tomorrow. Let's hop on, let's sketch this out. And it just completely opens the door to the viewpoints and expertise that you can immediately access.
So one last question, and we're going to close out, and then I'd like us all to move outside and continue to mingle and chat about this. But while we hope there won't be a next time, what have we learned for next time?
I think two things for me, at least. One is that we certainly can rise to the occasion. The development of the mRNA vaccines is probably the greatest biomedical accomplishment in the history of humankind. But I also hope that we also recognize why it's important to study interesting things for the sake of interesting things. I was an MD PhD student at the University of Pennsylvania. When Drew Weissman was there and Caitlin Carrico were there, they were just working on mRNA stability because they thought it was interesting. And I know there was sort of pie in the sky, yes, for my grant, I'll say it could be used for X, Y, or Z therapeutic, but they were really just interested in how to make a RNA molecule stable in a cell. And it wasn't sort of the quote unquote hottest work that was on campus, but they stuck with it because they were really interested in it. And there's something to be said about studying things that you're super interested in, because you never know what's going to be relevant.
I think I would say one of the great things about MIT is the benevolence of the donors gives us the flexibility to pivot, right? If we were somewhere else, even if we had these ideas, you need a little bit of capital to be able to make that transition. And really, MIT has been fantastic at working to provide ways that we could make that transition from what we had been working on to COVID. And I think that's really, that's a unique feature, of MIT, a place that's really designed to solve problems.
So I'm going to put my kind of educator hat on and teacher, which is, I've said for 10 years now, and this is as a biological engineer, but you could say that about many areas, obviously including life sciences. But I said there's never been a better time to be, a biological engineer than there is now, because we've never had an opportunity to make a bigger impact. And you could substitute many different disciplines in for that. And so, but people understand that. What I really hope is that that young people, elementary school kids and high school kids and college young people will realize what a great profession it is, and yeah, it is doing things you're interested in. Before I used to say this is the coolest job in the world because I get to study and build and figure out how things work. But now you have this really big link to, you say, and you can have a big impact on the planet. And I'd say that that to me is a tremendous event that comes out of this, to realize that people in this lab, these labs here, and the young people coming up that choose this can demonstrate and see what an impact they can have on people's lives and the planet. So I'd say that's a good thing that came out of it.
Well, I think that's a beautiful way for us to close out. I'd like to thank you for joining us and telling us about your journey through the pandemic. I would like to thank the amazing team who pulled this event together this evening. Thank you. I'd also like to do, a special thank you to our donors who have made so much of this research possible tonight. Some of this work was personally funded by some of our donors, so I want to thank you all personally. And I'd like you to join us outside for a drink and some nibbles. And thank you. And please be well and please be safe.